Hello everyone, welcome to the channel Dr. Hasi Explains. This is Dr. Hasi, and in this video I'm going to cover actually three topics: chi-square test analysis, Mendelian genetics, and then mitosis meiosis for the exam 3. Okay, let's start with chi-square test. So chi-square test, which is also written as x square. Here is the formula you see here in this very top of this document, which is chi-square is sum of observed minus expected square divided by expected. Now, so sum of these. So we always start with hypothesis. So look at the question and make an HO or a null hypothesis if data follows 3-to-1 ratio, for example. Then we make a table like you see here is on this document. So in the first column, actually we call phenotype. Then observed column is always given to you. Expected, you need to calculate. Then observed minus expected square, observed minus expected square divided by expected. So when you do that, you basically you add these together and you find a chi-square cal, which is the value we need to compare with chi-square table value. In order to find the chi-square table value, which by the way you never need to memorize, you need to know degrees of freedom, DF. DF is always DF equals n-1, n is number of phenotypes. If you have 5 phenotypes, your DF is 4, for example. And you go and read chi-square table value for that specific DF, and that's actually that number is very important because we need to cover and compare that number with our chi-square cal. So chi-square cal, we always want smaller than chi-square table value in order to accept this hypothesis. Okay, so if we accept it, then our conclusion is yes, data follows that 3-to-1 ratio, whatever we're testing. But if we have to reject it, then actually data don't follow that ratio. And then that's our conclusion, but we need to probably repeat this whole thing in the lab. Okay, so this is chi-square test, chi-square analysis. So make sure that you bring a calculator and then you do the test, and then make sure that you carefully calculate and compare these values and find your conclusion, chi-square analysis. Okay, so next is basic Mendelian genetics. Mendelian genetics, always it's good to start with actually definitions. What is crossing over, meiosis, diploid, haploid, dominant, recessive, fertilization, gamete, genetics, genotype, phenotype, haploid, diploid again, homologous, homozygous, hybrid. So it all starts with Gregor Mendel in 1850s and his experiment actually with garden pea plants. And when he crossed them with 2 different flower colors, you look at the F1 and he crossed again, he looked at the F2, and he came up with these, what we know Mendelian genetics today. And today many scientists actually do crossings to actually find better flavors of apples or disease-resistant tomatoes, and etc. Many areas. So make sure that you actually know phenotype always equals genotype times environment. Phenotype is it can be actually wild type or it can be mutant. Genotype can be homozygous or heterozygous. We always name genes with a letter like A, B, C. Big letter always means homozygous. I'm sorry. Big letter always means dominant. Little letter always means recessive. So, and it could be homozygous, same letter or different letter like heterozygous. Recessive doesn't mean actually it's anything wrong with it. Recessive means it only shows up if there are 2 copies of it. Otherwise, it doesn't show up, but it didn't disappear. Okay, these are genotypes like letters, right? A's, B's. So make sure you actually study from the book, our lab book that we did actually in the lab. Remember the genetics lab, Mendelian genetics lab, and lab 13, all the definitions there, and then little problems we worked, and then little Punnett squares we did actually to find genotypes or gametes from genotypes or genotypes from the gametes, vice versa, and also small problems we did. So to make sure that you can do these, and then you are good in this genetic section. So and then last lab, well, was not in this order, but last lab in this specific module is mitosis and meiosis. So we know mitosis and meiosis, they are both part of the cell cycle. Mitosis only happens in regular cells. Meiosis only happens in gametes, but they have a lot in common. Meiosis has 10 steps. Mitosis have only 5 steps. Meiosis is very important in genetics. Mitosis is just basically copying and making exact copy of a specific cell, is exactly duplicating it, is exactly identical to the two identical cells, two parent cell. But in meiosis, we have 4 offspring cells, 4 daughter cells, I'm sorry, and they are not identical. They are actually half the chromosome number of the parents. So make sure you study these 10 steps, and then make sure you understand meiosis is very, very important. And the daughter cells created at the end, they are not identical, but they are unique. That means they are unique. They are not exactly same as each other, or they are not exactly same as the parents. So we will so go over your notes and look at your book and keep studying. We still have enough time actually to do more review, and let me know if you have any questions, or just reach out inbox from the campus. Thanks for watching, and best of success in your last module exam this week. Don't forget to subscribe to the channel.